Please subscribe the channel and don't forget to click the bell icon.
Bismillahirrahmanirrahim. Welcome to Botanica. So in the previous section, in the previous class, we have discussed about and introduced you with the phloem transport, why the transport process is important in the plants, how the xylem and phloem are responsible for the transportation of different minerals, water and the food which is formed during the photosynthesis. And after that we have discussed about the experiment, an elegant experiment which was carried out by M. Malfigi, which is known as the girdling experiment, that he removed the bark of the tree and allowed the xylem intact, and what happened, there was a swelling, or we can say that the characteristic swelling occurred on the top of that particular girdled stem, which was the first line of evidence. We can say that the transportation tissue, or the tissue which is responsible for the transport of food synthesized or the photoassimilate, is the phloem. After that we have discussed the second experiment, that is the aphids, which was carried out by the insect nutritionist or entomologist, and that was carried out on the aphids, that aphids feed on the phloem tissue, that they insert their stylus, the long mouth part, into the phloem tissue, and they suck the food, or they suck the phloem sap, and after that, when the stylus was filled with the phloem sap, the aphid was anesthetized, and after the composition of cell sap was checked, analyzed, and it proved that their phloem is responsible for the transportation. And now we are going to discuss the third line of evidence, which is in the history, that and these three lines of evidence are to complete, or to say that, to confirm that the phloem tissue is responsible for the transportation. The third line of evidence involves the use of radioactive tracers. As in the plant physiology and biochemistry, mostly these are the radioactive compounds, radioactive tracers, which are heavier isotopes of those compounds, and which are used in the various experiments to trace the path of that particular compound. For example, we are using heavy nitrogen and heavy carbon in the photosynthesis in the Calvin cycle to check to check the trace and to trace the path of the carbon which is fixed from the atmosphere. And what is the reason of using these radioactive tracers? Because these radioactive tracers can be traced and can be distinguished by the normal carbon or the nitrogen compound or the normal atoms of that particular molecule. For example, we have a normal C12, which is and there will be a diversity of carbons which are present in the that plant, and all those carbon will contain C12, because in the atmosphere carbon is present in the C12 form. But here we are feeding, or we are giving a heavier carbon, so that it can be distinguished from the rest of the carbon which is present already in that particular plant. So to investigate and to check this, the third line of evidence, it is involved, and it is related to the radioactive tracers, and predominantly the C14, carbon 14, which is a heavier isotope of the carbon, or C12, and it is fed to the leaf. So when we are feeding carbon 14 to the leaf, a typical example is the translocation of photoassimilates in petioles of sugar beets. So this particular experiment was carried out in Beta vulgaris, and in these experiments, attached leaves which were attached to the plants, and they were able to conduct the process of photosynthesis. So they were allowed, and they were allowed to photosynthesize, or they were allowed to fix the carbon in a closed chamber where the only carbon source was CO2-14. So they were allowed to photosynthesize in a chamber, and in that chamber there was no carbon 12, but all the carbon which was present, that was heavier carbon 14. So the plant, whenever plant was able to photosynthesize, so that in the Kelvin cycle the carbon which is fixed, it will also be C14 and it will not be C12. So when C14 is fixed in the Kelvin cycle, triose sugars are formed. So what will happen after when the triose sugars are formed? So now we will be able to trace the path of this particular CO2. So through which of the tissues of the tissue system this particular C14 will move. So we will be able to trace the path of that C14. So after 10 minutes of what? After 10 minutes of photosynthesis in the C carbon 14. So the radiolabeled carbon dioxide or the carbon photoassimilate being transported out of the leaf. So after 10 minutes plant was able to conduct the light reaction, and after the light reaction the carbon molecules were fixed, CO2 was fixed, and that CO2 which was fixed, as there was only C14, so it was the radiolabeled photoassimilates. So the photoassimilates which were formed by the process of Kelvin cycles, that was also containing the carbon 14. So they were when they are formed, immediately they were transported out of the leaves and they were immobilized by freezing the petiole in liquid nitrogen. So when they are formed in the leaves, so they have to be mobilized by the plants from the leaves to other parts of the plants. So when they are transported from the leaves to the petiole of that leaf, so immediately that particular tissue, that particular leaves was freezed by using the liquid nitrogen, and liquid nitrogen has the temperature of minus 196 degrees Celsius, and it has a rapid freezing action. So whenever we put any of the plant organ in the liquid nitrogen, so that organ is quickly freeze. So that particular photoassimilate which was formed during the process of photosynthesis by the Kelvin cycle, that was immobilized, or it was restricted for mobilization. It was restricted to move from the petiole to other parts of the plant, because in the petiole the transportation will occur or will be occurring through the through the tissue, and we are concerned with to know that particular tissue in which the petiole that is responsible for the transport. So liquid nitrogen result in the freezing of that particular petiole and the leaf, which result in the cross section of that leaf, petiole of the frozen petiole were prepared and placed in a contact with X-ray film. So after when we have made any radio labeled cross section, so now we are putting that in X-ray, which will result in the resulting image on the X-ray film, resulting image of these cross sections which were of the frozen petiole, and it was of the petiole which was carrying out the leaf, carrying out the photosynthesis in the C14 environment. So the radioautograph indicated that the radioactive photoassimilates being translocated out of the leaf was localized exclusively in the phloem. So what they noticed, that particular C14 or the carbon 14 which was fixed by the process of photosynthesis, and the photoassimilates which were made, they were primarily located in the tissue, and that tissue was the phloem. So all these three lines of evidence, they suggest or they confirm that phloem is the transporting tissues of the plant, and it transports the food which is formed during the process of photosynthesis. So similar experiments of using the radioactive radioactive atoms, they were conducted on a variety of herbaceous and woody plants and with other radioactive nuclides. So there was many lines of evidence, but these are three just historical events which were used or which were able to confirm the path of photoassimilates from the leaves to other parts of the plant. So such as phosphorus and sulfur are also used with the same conclusion. The translocation of the photoassimilates and other organic compounds over long distance occur through the phloem tissue. So this is not only the case with the carbon, but also there are some compounds which are also transported through the phloem, and those are the other compounds which contain the phosphorus, and also they contain sulfur as well. So these both were also used to trace the path of food molecules which are synthesized in other leaves and other parts of the plant, and then they are translocated. So this was the little about the history of translocation of the phloem transport. What are the historical events, how this was confirmed, or it was confirmed that phloem tissue is responsible for the transportation. So when it is confirmed that phloem is responsible for the transportation, for the transport of food from leaves to the other parts of the plant, or we can say that from source to sink, the phloem tissue anatomists were concerned with the anatomy and the composition of the phloem tissues. What are the different types of cells which are in coordinatively or in combination they form the complete phloem tissue. So after the very elegant experiments, it was come to know after the anatomists, they had confirmed that phloem tissue comprises of the sieve tubes, companion cells, fibers and parenchyma. So these all are the four types of cells. Majorly there are the sieve tubes which are responsible for the transport. Companion cells are associated with these sieve tubes elements. Fibers and parenchyma are also associated, are also the part of this phloem tissue. So in combination, these four cells, when they are in the phloem tissue, they are responsible for the transport of photoassimilates. So a sieve tube comprises a system of cells where each cell is called sieve tube member. So as far as the sieve tube, as we have said that sieve tubes are the major elements of the transport, major cells through which the transport occurs. So sieve tubes comprises, they are the system of cells which are joined end to end. So one sieve tube is connected to the other sieve tube element, so they are making a complete straight line which are joined end to end, like you have seen in the spiral gyra algae, which is in which the cells are joined end to end and they are making the filament. One end is joined to the other and they are making the complete filament, lengthy filament, vertical filament. So these cells are similarly like that particular filamentous algae, spirogyra, and these ends are joined, and each of the member or each of the cell is called the sieve tube member of sieve tube units, or it is also called as sieve tube element. So remember, we are talking about each member, each cell. So each cell is sieve tube element or sieve tube member. So a sieve tube element is a thin walled elongated cell having living cytoplasm but no nucleus at maturity. So as in contrast to xylem tissue, phloem tissue or the sieve tube elements, they are living almost, and they are opposite, or they are different from the xylem, as they are living and they contain the cytoplasm. But there is also another thing which is important in the sieve tubes, that they have no nucleus at maturity. So they contain, first of all, the sieve tube element, they are thin walled, they are not the thick walled, and they are elongated as they contain the living cytoplasm. But they have another exception, that they have no nucleus when they are mature. The nucleus is present when they are young, or when they are dividing, or when they are completely maturing. So immature cells of the sieve tubes, they contain the nucleus, but when they are mature, the nucleus and the other organelles, most of those organelles are lost, and the mature sieve tube elements, they are without the nucleus. So this is the composition of the phloem tissue. You have seen that we have a phloem from this side, and we have phloem, parenchyma cells. These are the companion cells, you can see, which are associated with the sieve tube elements. And here we have also the phloem parenchyma cells. These are the sieve tube elements, parenchyma, plastids and other. So from here you can see that we have discussed about, we have a sieve tube. This is the sieve tube element, one sieve tube element which has walls or sieve plates, which are called because they are perforated. These contains the pores, so that's why these are called sieve plates. And one interesting thing, that these sieve plates are not straight but they are oblique. So this particular sieve plate is oblique, as well as this sieve plate is also oblique. It is not straight. So along this is the one sieve tube element. So adjacent to this sieve tube member, so this is, and there is another sieve tube element, and from this there is another. So here we have 3 sieve tube members which are joined end to end. This is the complete one sieve tube member. Adjacent to it there is another, and to the lower side there is also other sieve tube element which is jointed. And these all sieve tube elements are separated or distinguished by these sieve plates which are perforated and which have pores in it. So the movement of molecules can occur here. So along with these sieve tube elements, there is one important type of cells which are called the companion cells. Why they are called the companion cells? Because they are associated with the always with the sieve tube elements. So that's why they are called the companion cells, those cells which are always associated with the sieve tube elements. Whenever sieve tube elements will die, the companion cell will also die, as it is associated with the mechanism, or it is associated with the action of sieve tube elements. Along with the sieve tube elements and companion cells, there is the Phloem parenchyma cells which are also associated in the tissues, and they have also their parts. So these are the sieve tube plastids are also there in the sieve tube elements, and parenchyma plastids are also present here. That there is a nucleus in the parenchyma cells, Phloem parenchyma cells, but you can see here in the mature sieve tube element there is no nucleus. So as different to the other tissues in the other cells in the phloem tissue, these sieve tube elements have no nucleus. So more we are discussing about it in the description. So the distinguishing feature of the phloem tissue is the conducting cells, which are the sieve tube elements or sieve members, which are responsible majorly for the transport, and they are also known as the sieve tubes. The sieve element is an elongated rank of individual cells, as you have seen in the diagram, in the picture, in the figure, that these are longitudinal and they are elongated as they are jointed end to end, and these are called sieve tube members and arranged end to end. So unlike the xylem trachea elements, phloem sieve elements lack rigid cell walls. So as these are the differences between the xylem and the phloem. The xylem have the rigid cell wall, but the phloem are the sieve tube cells, which are the major transportation cells. They have the thin cell walls, and they contain also living protoplast when mature and functional, but when they are non-functional or when they are immature or when they are in the developing stage, so these things can be checked. So the protoplast of the contiguous sieve elements are interconnected. Contiguous means those cells which are jointed end to end. So in this diagram you can say these are the two contiguous cells. So the cytoplasm of this particular sieve tube element, and these are jointed as end to end by the sieve plate. So these cytoplasm are interconnected. So the protoplast of contiguous sieve tube elements are interconnected, and these are the sieve areas, or these are the sieve plates. So where the pores of the sieve area are relatively large and are found grouped in a specific area, they are known as sieve plates. So the cytoplasm of these sieve tube elements is joined or is connected with one another. So they are making a long continuous rank of cells which have the simple one. We can say that one cytoplasm, as it is connected with one another, so they are not different cells, they are not separate cells. So that's why we can say that. So sieve plates are typically found in end walls of the sieve tube members and provide a high degree of protoplasmic continuity, as they are responsible for giving the continuity of the protoplasm. So in addition to the sieve elements, phloem tissue also contains a variety of parenchyma cells. So along with the other cells which we have discussed, the fibers and other cells, so there is also parenchyma cells which are also called as the companion cells. So some of these cells are intimately associated with the sieve tube members, and these are called the companion cells. And we have discussed why they are called the companion cells, because they are associated only with the sieve tube member, and when the activity of sieve tube member is not more required or they are dead, so companion cells also die and they are no more there. So that's why their life is dependent upon the sieve tube elements. So that's why they are called as companion cells. So contain and in contrast to the sieve tube element, they also contain a full complement cytoplasm and cellular organelles, as there is no nucleus and other organelles in the sieve tube element, but parenchyma cells has a full complement of cytoplasm. It also contains the nucleus, and it also has the other organelles which are required for the proper cell functioning. So interdependence of the sieve tube members and companion cells is reflected in their lifetime. As we have discussed before, that companion cell will be alive only when they are, so long as the sieve tube members continues to function. So when the sieve tube members die, its associated companion cells they also die, and companion cells are believed to provide. So that's why, as these companion cells contain the complete nucleus, they contain the nucleus and other organelles, so they will provide the metabolic support, and they are making everything which is required for the proper functioning of the sieve tube elements, and perhaps are involved in the transport of sucrose and other sugars into the sieve tube members.